give us 2 minutes for the recording to start. Yeah, ma'am, please start introducing.
Thank you, Mr. Majid. A very good morning to all dignitaries and participants. Today, the duty invested in me is to invite one of the very eminent and vibrant scientists of IVRI Bareilly, Dr. Sanjay Kumar Singh, Principal Scientist in the discipline of Veterinary Gynecology and Reproduction. Welcome you, sir, to this 21 days training program.
Very good morning.
Sir, let me introduce him with a brief introduction. Sir currently is working as the in-charge Animal Gynecology Laboratory and Institute Technology Management Unit, Zonal Technology Management Committee at ICAR-IVRI Izatnagar and has more than 21 years of experience in research, teaching and training with expertise on assisted reproductive technologies, primary cell culture of utero-ovarian origin, molecular reproduction, diagnostics for improving fertility as well as infertility, and obstetrical problems in livestock for enhancement of reproductive efficiency, then IPR, technology transfer, and ABI. Sir has handled a total of 7 institute-funded projects and 6 external funded projects, and developed a number of technologies which include development of 2D endometrial and ovarian cells culture in buffalo, designing and synthesis of multiple antigenic peptides specific to pregnancy, transcriptional profiling of oxytocin, OTR, prostaglandin, steroid pathway genes during uterine infection, cyclicity, pregnancy, and formulated mineral supplement for buffaloes, development of goat pox and PPR vaccines, devised anti-luteolytic strategies, etc. Sir has filed 4 patents and including live attenuated vero cell based goat pox vaccine for protection against goat pox and formulation of mineral supplement for cattle buffaloes reared under wheat rice sugarcane farming system, and 2 patents on synthesis of MAPs against protein specific to pregnancy and their use in EPD, and awarded 1 patent on goat pox vaccine. Sir has also associated as inventor of the team member for goat pox and PPR vaccine technology commercialization to many industrial houses and realized huge revenue to the institute that amounts more than 4 crores through license fee and royalty. Sir has also filed 4 designs related to reproduction, and 2 of them have already registered at the Indian Patent Office for design in addition to 4 copyrights on critical care of diseases, technology services, apps, AI, etc. Sir has received many awards, fellowships and recognitions including DBT Crest Award, ICR Team Research Award, Niels Lagerlof Award, Young Scientist Awards, Dr. S.N. Luktuk Award, Best Poster and Presentation Awards, and currently he is working as Associate Editor of JHP Nexus and member of NABS, BOS of Argo discipline of BHU, Varanasi, etc. Sir has also visited Nottingham University, UK under DBT Crest Award for 1 year advanced research, and he has received postgraduate diploma on technology management PGDTMA. Sir has published more than 73 research papers, including national and international recognition, and written many book chapters and published a number of popular articles and leaflets and folders, review articles, manuals, compendium, and guided almost 5 PhD students and 6 MBSc students, and he is member of the advisory committee of many students. With this brief introduction, I am welcoming you, sir, once again to this 21 days program to deliver his talk on the topic of early pregnancy diagnosis, a tool to increase reproductive efficiency in bovine. Welcome you, sir.
Yeah, thank you, Dr. Devi.
Thank you,
I'm really, though I think I don't require, but you have asked such type of the biodata, but even then I'm happy to at least you have given a glimpse of my personal and academic profile. Thank you very much. So should I start now?
Yes, sir. Yes, sir.
Okay. The slides are visible now. No issue.
No, you have to go to the slides.
Okay. Yeah, it is okay.
Yeah, can you press the hide option at the bottom?
Okay. Good.
Can you start in full screen? Can you press F11, I guess?
F11. Yeah, it's okay.
Yes.
Thank you all, and very good morning to all participant, trainee, whatever you can say. You are the wisdom of our, future, past, and present everything, because you are the main asset. So Dr. Sudesh, most respected Dr. Sudesh, Dr. Gopinath, Dr. Rinku, Dr. Majid, and all the trainees. A topic which has been assigned, I think, I don't know how much relevant to all of you, but I think I asked Dr. Sudesh, I think around out of 150, something like that, around 20 are from reproduction. So no doubt, I think for others also I have tried, but whenever you feel it is over, you can ask me, sir, you can stop here or something like that. I don't have any issue, because I am trying to cover whatever the state of art is there, and further what we have done in the recent past, as well as since long I am working in this particular area from my PhD. Then again I had a break, I went to Mukteshwar, then again I started searching some other options, then again came back, again it started. So this area I am working since 1995, and now I think I am very near to reach the target. So that I am going to share the topic actually Gopinath has already told, early pregnancy diagnosis. And so the reproduction, I mean, it is reproduction is luxury, and reproduction will happen when the body is fit. So that efficient reproduction is a vital component. Same is true in humans, same is true in the animals. Is a vital component of livestock development program and its success. And reproductive efficiency, can you say in other word, maximum use of the reproductive capacity of the animal is of considerable practical as well as economic importance, and it can be enhanced to a certain limit by modification in the traditional methods of breeding, feeding, and management, because they have tried with one or more angle. And genetic selection and scientific management practices, undoubtedly it has increased the milk yield, but simultaneously it has compromised the reproductive efficiency. And in general, in an 20-30% of dairy animals, they are being culled because of infertility. That is a major concern, and therefore higher reproductive efficiency, it requires orchestrated management activities, especially at the age of calfhood, heifers, and first 100 days after calving, integrated with advanced technological intervention. So this is the crux, whether we can definitely increase the reproductive efficiency or not. So in general, if you see the poor reproductive efficiency in terms of reduced milk yield, increased cost associated with the multiple insemination, increased number of the replacement heifers needed to maintain the herd size, altogether they are major factor which affect the reproductive performance. And even a one day increase in a day open period, it is around the loss of 281 to 360. In general, if you say it varies from cattle to buffalo, even roughly 250 per day, one day extra cost. Just you imagine one cycle, just you imagine 2 cycle, 3 cycle, and in the field setting situation is quite adverse. So therefore, an early and accurate diagnosis of pregnancy, it is definitely a requirement of this country. Abroad so many things are there, but still I couldn't say everything is so perfect and people are having all those state-of-art technologies for early and accurate diagnosis of pregnancy. So it will be a tool for reproductive performance as well as productivity. So this is definitely a goal. If you agree, getting a calf at every year 12 to 14 months in a cattle and 15 to 18 months in a in buffalo, say 2 calves per 3 years in buffalo, more rightly. It is definitely is a vision, it is a goal, it is everything. It is, for as a reproductive biologist, and these are the certain reproductive efficiency indexes are there. They are very important. If really you want to achieve this goal, you have to work on these areas. And that is very important. So whatever the technology advancements are there, we have to integrate it if really we want to improve the reproductive efficiency in the livestock. So there are 2 basic approaches. Either we should increase the fertility. If you see where the field and farm settings, people say fertility is hardly 30 to 40%. So can we increase? So that, and one approach should focus on the increase in the fertility. Another is the infertility. I have rightly pointed out around 20-30% infertility cases are there. Can we intervene at some point so that we can reduce the infertility, and that will altogether will increase the fertility? So just a brief background as far as pregnancy diagnosis in Bovine. So it is there. Just this slide itself is a very well explanatory. I'm not going to all visual and all those conventional methods. The methods which are being used still on date, and in that series, rectal palpation is the first and foremost method. If you see the books, and people say it can be used very nicely to 35 to 45 days with a skilled person. One word I have used, skilled person. So how much you are skilled in this art, that affect how early you can diagnose through rectal palpation. But in general, at field level, I will suggest, and I think most will agree, the 60 to 75 days post conception is the right time to have an early pregnancy diagnosis. I mean, through rectal palpation, not say early, pregnancy diagnosis through rectal palpation. So, and even then 60 is a good days for cattle, and 75 days because little, if you say the embryonic development is not as fast as in case of cattle in Asia. So I will prefer 75 days post conception. If it is heifer is there, you can take 10 days less, means 45 to 50 or 60 to 70. If it is a terrace animal, definitely 60 to 75 days post conception in Indian setting, whether it is a farm or clinical field setting, I think it works. Then people they thought what other things can be done in the meantime. Some laboratory methods like measuring the blood and milk progesterone that says 21 to 24 days. Around 1980s, people they started thinking yes, it is a very good test. Yeah, undoubtedly it can diagnose pregnancy with 100%, no issue, because it is all related with the presence of the corpus luteum and that secret progesterone. But the moment when you say how much pregnant animals they are, so again, I mean, a lot of wide variation is there, but still I won't go beyond 60 to 70%. But there are reports depending upon number of these, I mean, size and way and type of the kits, methods adopted. People they claim 60 to 90%. Altogether, still the sensitivity to diagnose, I mean, pregnancy is really a challenge. So people they start thinking some other areas. Ultrasonography introduction in the animal reproductive science is though fairly recent. From 2000 onward, people started thinking, yes, we should introduce this in our heart or in our settings. And people say, I mean, as far as the reports are concerned, it can diagnose a pregnancy with around 100% success, 90 to 100%, you can say, by 28 days post AI. But it requires specialized equipment, trained person, and I hardly believe more than 5% are trained in all these settings across the universities, institutions, field. More than, I think, I don't find 5% they are trained in this area. But we do require, because if we have to work, we have to take all the methods all together depending. So as far as all these methods, they have the potential. Undoubtedly, they have the, I cannot challenge this is good or bad, but it is you who has to decide type of the animal situations, where you are working, I mean, accordingly. These methods, they have the potential, but definitely they fall short of expectations, being as a practical on-farm use for early pregnancy diagnosis, which emphasizes that there is a need for early and accurate pregnancy tests well before 30 days post-calving, with the minimum physical handling, and it is responsible for sound reproductive health, and it is going to, in other way, it is going to save high percentage of concepts which are lost before diagnosed by conventional methods. So, just a brief, because no, I'm not going to touch ultrasonography, rectal palpation. Everybody know all techniques. Just ultrasonography, I will little touch. It is a non-invasive, you can say semi-non-invasive, immediate and safe evaluation. High frequency sound waves are there. Different frequencies are there. As far as diagnostic applications are there, and its basic principle, you know, all I think everybody might be knowing. It is a pulses of ultrasound which are generated by the piezoelectric crystals in the transducers. A different type of the mode are there, A mode, B mode, and the type of the instruments, if you see, as far as transducers are concerned, two transducers are very popular, linear and sector. And linear transsector transducer is very common for large animal and ranging from 5 to 10 megahertz. So linear, and it gives a rectangular shaped image. However, when you say it is sector, it is a pie shaped image. So in cattle and buffalo reproduction, linear array, real-time beam mode ultrasound, they are the best scanner. And if you say how you visualize the uterus and conceptus, I mean, initially while working with this ultrasonography, I'll suggest at least you should at least scan 100 animals which are non-pregnant at different stages. Don't think about pregnancy first. And once you are able to visualize all those uterus and other structures, related structures of the reproductive organ, then very nicely you can diagnose pregnancy at an early stages. And imaging of the bovine uterus, and it is cyclic curve because uterine tissue, they appear like a echogenic structure on the screen. And uterus is comprised of soft tissue. It absorbs some part of the ultrasound waves and that reflects a portion of the waves. So normally it appears gray structure on the urine. It is clear. Any doubt, any feedback, because it is a one way only, I think. But any one feedback, should I go ahead, or it is I mean, you are able to understand? Is there any issue, Dr. Naz? Any comment? Yeah, thank you very much. Okay, thank you very much. So nice of you. Yeah, so once we see the cross-sectional versus longitudinal section of the uterus, yeah, a cross-sectional of the view of the uterus is displayed as a rosette and easily distinguished from other peripheral structures. And if you say longitudinal section, which is less will appear as elongated structures. So it depends. I mean, those who are working in this area, they can know how ultrasound works. And people say, again, there are a lot of debates are there, a lot of variations are there, how early. But people say it is 9 days also, 10 days, 12 days. But when you can go for early, but saying early is not important. Saying how much sensitive the moment when you go beyond 90% to 100%. And that accuracy reaches after 25 days. So basically, we should have two approaches and while using ultrasound. First, emphasis must be given to identify non-pregnant. I told you, you must be an expert first to declare non-pregnant, so that you can take all those care what non-pregnant are supposed to get if a management strategy can be adopted. Then pregnancy comes, how quickly you can diagnose the pregnancy. So this is a very milestone paper developed by Quran and co-workers in 1986. You can go through all these papers. These are the different characteristics. When you can palpate all these structures, they have different range. So they have started as early as from 19 to 55 days, something like that. So all ultrasonography, it lies between this particular period. So depending on some of these are the visuals which we have done in our laboratory settings in experimental system. These are how the buffalo follicle look like, how the CL look like, and these are pregnant uterus you see. So this is fluid at around 30-35 days structure is there. Some other images, day 25 images, day 30 you see embryo proper, day 35 days. Some again embryonic vesicles, how the heart look like, how the embryo look like, embryo proper you see. Some very good photographs are there. Those who are in this area, they know how things are look like. So just I will showing you all these. And further also you can see scale, you can measure diameter. Now machines are very advanced. Once we were working around 20 years back, machines were very primitive types. We have used pi medical machines, I think, quite old, but slowly, and now machines are very good. And so as far as accuracy, sensitivity, specificity are concerned, it is a reliable using a linear curve with measure pi 5 to 7.5 depending upon the type of the machine. Accuracy does not go beyond 50%. I have rightly pointed out just like it between 16. But the moment in field condition, I recommend that you should use ultrasonography by 25 to 26. The major reason that at least animal must have passed one cycle, so the chances of errors will be less, and you will be also more confident, and farmer will be also more confident. So before that, incorrect diagnosis will be definitely high. So accuracy is very good when you go beyond 25 and 26. 28 and 20 to 30 days is a very ideal situation as far as ultrasonography is concerned. So you need not to but even then one repeat examination, if you are working at field system, one repeat examination I will suggest you should do again after 15 days, so that and even in human also you see after diagnosis, suppose 7 day you did a video kit, SCG kit, and then again you went to doctor, and then doctor started showing yes, no ultrasonography also. So now you come every, so almost every month the ultrasonography will be. So in animal system also, we should not rely just once we have done it is over. If you can pay and you know your animal is very important, at least periodical testing is very important initially with ultrasonography or other way of laboratory methods, followed by rectal palpation or something like that. So that you must be ensured throughout these stages of the pregnancy, yes, my animal is pregnant and I should take care of according to that. So I know color Doppler has come, and you can visualize all those blood flow. These are all advancement, and I have told you all these very nicely. You can achieve the accuracy of more than 99% at around 28-30 days. Sometimes I have told you subsequent examination you should do, and in addition, I have not covered, but USG is also helpful in diagnosis of various forms of infertility, especially ovarian disorder, and especially in pet cases. That is not the domain of this particular lecture, but those who are expert, they are knowing well. But major issue is that you must. I have seen many ultrasonologists in human also. They say they are doing ultrasonography, but I think they are just doing for the sake of ultrasonography. The kind of diagnosis, the kind of expectations which we do expect from ultrasonologists, that is definitely missing. That's why you must be really very skilled as far as ultrasonography is concerned. So, and now, another method everybody might be thinking, sir, till now so telling whatever is already known. But many things are known, but I have tried. But the laboratory methods, yes, they are the big bet in this particular field. And initially, I have told you the method has been developed by measuring blood or milk progesterone, and more recently, people they are using a protein which are specific to pregnancy. Yes, definitely that will be a very specific, because these are the protein which are secreted by the conceptors. Whenever there is a presence of conceptors, only these proteins will be there. Therefore, such type of diagnostics they sounds more than the other method which has been derived based on the metal-derived substances like progesterone or others. So initially, I will just start with the steroid, and the corpus luteum secrete progesterone, and many other hormones are also come, estrogens like total estrogens. They are also helpful, and they are the indirect means of pregnancy evaluation. And so based on that, people they have used for pregnancy diagnosis invasive blood or plasma, non-invasive milk, saliva, urine, feces. So as far as progesterone are concerned, you see the most of the phases of a pregnancy stages, the progesterone concentration is high. That's why people they started thinking we must have a method which is based on progesterone. And another is total estrogen and prolactin, but they are higher during the later stages. So as far as plasma progesterone is concerned, and by Robertson and Sadra, they have described a method for diagnosis of pregnancy in cow by determination of the plasma progesterone concentration. And the variation as far as the peripheral plasma levels are there between pregnant and non-pregnant. Their clear-cut difference are there, and that difference people they have utilized for diagnosis of the pregnancy. Buffalo can also be diagnosed as non-pregnant by determination of plasma progesterone 21 days after insemination. Same is true in cattle as well as buffalo. And so I will suggest you at least one cycle must be passed, because a right period is between 18 to 24 days. 18 means MRP. If conceptus is there, animal has been, fertilization has occurred, embryos started developing, and MRP will be there. So MRP is very important. And if you see the MRP in case of cattle, it is around 16 to 17 days as per the short. And we have done a very good work on the MRP in case of buffalo. MRP in buffalo occur a little early, 14 to 16 days. So that's why we have put this particular window, 18 to 24 days post breeding, and that is a very nice breeding. Because as an invasive method, every time you cannot go for serum. People they have tried to use milk also, especially for the paras animal. This is not good, and you cannot do anything in heifers. When paras are there, milk progesterone is also a good way of getting the biological sample and doing the pregnancy diagnosis. So again, also optimum time is after 24 days after service. So milk progesterone determination is a practical method in tropical cattle as well as in buffalo, particularly for detection of non-pregnant. I have told you very nicely. If definitely you want, if you don't have any other facilities or laboratory methods, you can use this method so that you can separate all the non-pregnant animal, and pregnancy you can diagnose by other methods. Milk progesterone again, it depends on the type of the kits which are available, and SA, RIA or EIA or something like that. But in general, it is always more than 2 nanogram per ml in case of pregnancies. Cattle above it is more 4 to 5, and beyond that. Or in buffalo, it may be 2.5 to 4.5. If you see the Garvasu reports, you will find around 2.53, something like that. So the variation is there, but it depends how your assay behave. So In milk also, because it is a strand, so the milk concentration is high. So even if it is around more than 10 nanogram per ml. So these are the just way how people are using in the milk. It is a normal assay as maybe RIA, AIA based on the immune complex formation, and you have to detect that immune complex. Even some people they have also explored saliva. Now people they are start trying to explore all kind of the molecules in all biological fluids which are secret. Saliva is very easy. Most of the because ruminants they are ruminating. Most of the time frothiness is there. So people they have tried to measure saliva also, but the concentration is questionable. Urine also people they have tried to measure all this because in urine it converted to some metabolites, and people they have tried to measure these progesterone in the urine. And another area where especially for wild animals, fecal progesterone metabolites they are being used. Again, question is that it is very good for accuracy to non-pregnant, but when you say pregnancy, 67% to 70, and wild animal it is good. I will suggest, and even if you see most of the draft breed abroad also they are using these fecal metabolites, progesterone metabolites for diagnosis the pregnancy or something like that. And concentration is quite good, 15 nanogram per gram of the fecal material. And sensitivity is a quite variation. It depends lab to lab. You have to standardize all those things as per your lab at least, and accordingly you will be able to tell the things. So these are the some I have told you, progesterone and milk, plasma, and it varies. But think everything minimum it should be more than 1.5 ml or 2.5 nanogram per ml. So these are some of the commercial kits which are available based on progesterone. Kit by Polycam Hygiene Lab, Pune. This is an Indian-based kit. Another UBio also they are using a bovine pregnancy spot assay. But though you can say they are available, they claim also. But again, you see the sensitivity, specificity always it will be good. Sensitivity is a question, and they claim I don't know how much animals they have included there and what is. But if you see the commercial application, always it put a question mark. I couldn't find any all of these kits commercially used across this different setting, but they are available. You can see from Bio and others. The one very milestone work had been done by Tyagarajan and Padmavan long back, and then after that VD Lakchora from Panagar also they have used MAB. They have developed MAB and MAB based progesterone assay. But these kits have been commercialized by NRDC also. But I don't see, market. As far as market is concerned, I don't find these kits which are being frequently used by the users, end users. But they are available, which are based on the progesterone, and these are some of the progesterone-based kits which are available abroad with different name. Again, accuracy, sensitivity. I tried to cover almost all. Lot of variations are there. So as far as same thing, for specificity it is good, but the moment it comes sensitivity, it puts a lot of question mark. So the progesterone assay, these are some advantages I have told. You can go for early pregnancy diagnosis with good accuracy, but not perfect. You can say, but perfectly for a non-pregnancy. So there are a lot of limitations are there, cost and others. RIA, other issue, and if AIA is also there, you can use, no issue. But false positive cases are very high. So while you should try, as in human also, people are trying a number of the ways, number of the methods for pregnancy diagnosis. We should also use. I personally believe you should go for rectal palpation, you should go for ultrasonography, you should go for so that if the animal is primed, you can go. So now the area where we are working, and we're very pertinent, what are the proteins which are secreted by the conceptors and which are being used for the development of the diagnostic kits. These are very, since 1988, if in 1982, sorry, Butler and co-workers, they were the pioneer in this particular area. Though work started in 1974 by Morton and co-workers, and initially they have identified one molecule, early pregnancy factor. People made a lot of claim based on APF But again, when it comes in the real life situation, real life setting, it puts a lot of question mark. But definitely, this is a molecule which is secreted by the conceptus. How far it is good for as a diagnostic, that is another molecule was interferon tau. Yes, this is the first signal which conceptus secrete. Yes, we must be saved. We need some signals from the mother to protect us, and that is the interferon tau, and that is a kind of anti-lucrative nature. It stops the lysis of the corpus luteum, and accordingly then CL is maintained and the pregnancy is maintained. Another molecule by Ramu and co-workers, they actually the pre-implant factors. They have also, it's one paper basically, Ramu and co-workers, very good paper is there. They claim pregnancy can be diagnosed, but I couldn't find any commercial diagnostic kit based on that. Some other biological workers, they have tried to explore in milk and urine. I mean, but the major focus was in the serum. And I have told you, Butler co-worker, they have done a very milestone work in the discovery of the pregnancy-specific protein. It is definitely it is a new approach. Until now, many pairs they have been synthesized or sequenced, or they have been by different ways, biochemical or the molecular way. But some of the limitations are also there that I will discuss in the subsequent slide. I have told you this is the discovery of the PSBB in Bovine, and it exists in several isopyropha because it is a glycoprotein and it is secreted from the fetal cotyledon. It's a new approach to pregnancy diagnosis. Many patents have been filed, and this is the three most studied bovine packs. Which is, one is PSBB. Some kits are based on pregnancy-specific protein B, and the Sasser and Grove. Subsequently, I met these guys when I was in UK in Edinburgh. I had one conference. I had an opportunity to meet all those guys who have developed this. And then another is PAC 67, another PSB. Altogether, they are the same protein, but they are having a different N-terminal sequences, presumably which are identical upload-related proteins. So, just a brief more about that. These are secreted by the placenta of the all Aetherian mammals, which is including from cattle, buffalo, sheep, goat, and horses. They belong to aspartic proteinase superfamily and sharing about 50% amino sequence are into the cathepsin, cathepsin B, and cathepsin E. Function is almost very ill defined, but even then, whatever function has been defined, it has a multiple role in start from placentagenesis, fetal growth, fetal-metal communication, and immunomodulation. So they have been classified into two packs, ancient packs and modern packs, and you can read good very literature by in these areas are there. These are the secretary packs. You see how they are expressed. Different kind of the packs, they express different stages of the gestation. And same way, even as early day 7 also, there are many packs are there, 7 to 14. Usi Java and co-workers, they have done a very milestone work and using all microarray techniques, and they have told all these expression profile of these packs. Further, you see these packs, they are being secreted by two types of the cells present in the placenta or fetal membrane. One is binucleate cells and the mononucleate. There are certain packs which are secreted from the mononucleate or binucleate, but all those which are secreted from the binucleate cells, only they come in the circulation. Wherever most of the focus was on this particular group. So yes, I have told this Butler, and they have initially isolated and purified these two new proteins in the form of PSBB A and D, and D was found to be specific to placenta, and similar protein PSBB 60 and packs. Then further Bakers, they have purified bovine coronary gonadotropin, later identified bovine pack 2. So similarly, Jolie done a very good work. If you have to see the literature on these packs, you see this paper, Jolie and co-workers and antibiology of reproduction. That's a very good paper, you can see. I have told these all things already. So they are very heterogeneous in molecular weight, and the charges they differ in their amino terminal sequences. Molecular weight range, because I told you a lot of glycosylations are there with the glycoprotein, depending upon 37 to 78 kilodalton. And it all because of different degree of glycosylation. I told you till now 22 cDNA of packs, they have been cloned, fully sequenced. However, not all the packs have been purified so far, and the discovery of packs has provided a breakthrough for early pregnancy diagnosis research. These are the concentration of the packs in maternal circulation, and then you see how the concentration of the packs at different stages. You can see, and very nicely. I have covered almost this paper has covered almost all areas which are available in this particular aspect. I tried to cover, and the very if you see day 20 says, as early day 20, 18 to 20 it start, and then as pregnancy advances, the concentration goes higher. So people, they have tried to measure the PAG in the milk also. There are several factors which affect the PAG, like what is stage of pregnancy, breed, parity, body weight, sex of the fetus, placenta mass, half-life of the PAG, feeding. There are so many issues, and number of the issues are there. They affect, but they have certain promise, and based on that promise, assay has been developed. Are they utilizing the whole protein or group of the protein, or people, they have targeted immunodominant regions of those particular proteins, and based on that, ELISA or RIA has been developed. Significant variation again, as other assay also had, is between the assay, different ability is there to detect, and some of the cross activities also, especially postpartum pregnancy is also having some limitations. Then these are the another area where people are trying to have a linear or multiple antigenic peptides which are very specific to a particular protein, and there is a possibility they will eliminate the non-specific binding with a greater efficacy, sensitivity, and accuracy. So accordingly, people have tried to measure these protein in blood, milk, and urine, depending upon the type of the protein. And commercial kits are available abroad. Some of the kits are being marketed in India also. These are the different kind of the kits: Bioprint, Bovine test 29, IDEX bovine pregnancy test, IDEX milk pregnancy test, bovine pregnancy specific protein B ELISA kit, bovine pregnancy diagnosis BG29, Bovine, with different names are there. Especially IDEX is they are marketing the kits. In India, it is available in India. Cost is definitely, they are very costly, but some, I think NDDB and some organization, they are using. But again, we have also used, but I don't want to point out what is the, because this is not a platform where I should address what is the performance of these kits, but people are using, and these are the kits and their performance, sensitivity, specificity, accuracy, and all those. You see, sensitivity, people say all based on PAG, on PSB, they are having very good sensitivity, but EPF, and they are put a question mark. So anything beyond 90% is considered good. So at least they are safe, and people try to use. But in Indian scenario, we don't have much data, large number of the data based on 10,000, 20,000 animals using the similar kind of the kits. Only then we can say how far they are effective in our Indian scenario. So same thing, I think, is more elaborative. What are the companies which are making like ECF test based lateral flow conceptual diagnostics and Bioplane, something like that. Days are same, because they came very early. The moment is so very early, the sensitivity is questionable. But the moment it is day 28 and onward, it is good. So accordingly, there are, if you see Indian scenarios are there, the 3 recent reports occurs. One report from NDRI, they claim they have developed an LFA, but still, I think 1 year passed in one of its NDRI Kisan Dairy Mela. But this is the same group. We are also working. I am having a 1 DBT project where all 3 partners were there: ICAR, IVRI, and the NDRI. NDRI claimed that these kits in one of the projects they have developed, but still we are awaiting when they will see the market and the detailed performance. One group from Mumbai and others, a research team from Institute of Chemical Technology, Mumbai, and they have just put this in media. They have developed a urine-based pregnancy test kit. Same question: how far they are sensitive, how much they are specific, what is the data size. The third kit, which you might be having, only 4 or 5 months back, ICAR has released. It is also a urine-based calorimetry method by Balhara and group. They claim they can also diagnose the pregnancy. It is a test where they are taking a urine, adding these all reagents in a sequential manner, and they are boiling the urine, and it gives color, and accordingly you can take as the color, and then you can diagnose the pregnancy. Question is again same: how much sensitive and how much specific? But they are very good lead, and I hope in Indian scenario also, within I think within a year or 2, we will have definitely some solution in this particular area. For me, I think these are the 3 papers which really give me a lot of stimulus. I should work, because since long I was working on the biochemical aspect of these proteins. We were trying to biochemically purify it and all of those things. But again, after coming from the UK, I started exploring another area of that, and that the synthetic peptide biology, and accordingly we have focused our area in this particular utilizing this molecule. So initially work, I have told you, initially I worked, this is all my PhD work, and we have tried to biochemically purified packs, and we could success, but the time was different. We were not having much facilities as far as I personally believe, being a man of reproduction. But then I have been switched to Mukteshwar. Domain was different. Then again we came back. I joined in 2005 again to IVRI. Then we have submitted another project in NFBSRA, where one of my students again started working, and we could, Dr. Pradeep, he is posted at Hisar. He could also biochemically, he tried to purify it, and we could purify pack 7 and 11 in case of buffalo. So one of my student, Dr. Jerome, is also scientist at Hisar. He's cloned and characterized pack 1 gene, and crossbred cattle also I did at Mukteshwar. So these are very initial work. Now from 2014-15 onward, we started thinking of this particular area. Until now, we have synthesized 16 multiple antigenic peptides which are having role in pregnancy, establishment, and maintenance. And 4 MAP, they have been found very specific, which give a very strong reactivity. We test sample in the ELISA platform, and we have tested all these maps in the ELISA platform. And further, we have confirmed these maps through immunohistochemistry and the immunolocalization of these pregnancy-related AGL protein in the fetal membrane. It has shown that yes, these are the proteins which are secreted by the conceptors, and there may be a possibility it can be utilized for development of the assay. Accordingly, we have developed a sandwich ELISA in the test sample, and initial results were very encouraging. That's why we switched further. Though sensitivity was very less, but the higher specificity put has given a very good stimulus. Yes, we should think on that. And based on the initial observation and synthesis of the map, we have filed 2 patents. Now these both patents have been published also. So, and then we thought we should test more number of the samples, and further we proved work, and then we could see these are just how we have developed the sandwich ELISA, and we have developed now we have developed LFA also, and these are the performance. Now we have reached till now. So initially we had a direct sandwich ELISA, and we were achieving around 72% sensitivity and specificity. Then these were 2 milestones were done by Dr. Nitish Kharaya, who is scientist at the IIT Mukteshwar, and Dr. Priyaranjan Kumar, who is assistant professor at BHU. These are 2 students who did PhD under me, and it was there. I mean, it is a big stimulus for us, and they all worked in the DBT project. And earlier, the Institute funded project, and followed by DBT project. Then Dr. Amit, who is commanded in the BSF, he take this assay to further level, and we could reach to this. You see, 82 to 97. Again, he has given some scope to improve further, and then we are trying. He has developed both LFA as well as the sandwich ELISA, and these are the one of the plates how they look like. These are some of the samples. Whenever there is a pregnancy, we are getting test line and control line both, and when it is a negative, we are getting single. We have tried to put in the cassette also, and this is the way how it look like. So we have compared. I have told you very nicely. We are getting very good accuracy as far as ELISA is concerned, and same is LFA also. But definitely we have to work in this sensitivity. We are working now. These are very good number of data, around 200 ml. You see. So now we have switched over to more 400 ml data, and we have compared this with ultrasonography as a per rectal also. So this is with per rectal and ultrasonography. We have put this 70 to 35 days window with a very specific cause. By 17 days, MRT will occur. So there will be a non-specificity. I mean, false positive will be less. So that's why otherwise this is the protein which we have deleted as early as day 7 also. So, but for the convenience of this system, for the more success, we are putting this particular window or detection window of 70 to 35 days. And again, we are trying, because you know the ELISA normally being done in 2 days. And what we are trying to develop the ELISA in 1 single day. And another also attempt which we are making, we don't want to use any incubator, anything. Only simply at room temperature, and just having a for colorimetric assay, because it is a color clear direction. We require a laser result, and normally if you see in the fields or field setting also very cheap and cost effective ELISA results are there. So in that intention, initially we had our conventional assay. Now we are trying to improvise this assay and doing at 37 and room temperature in a single day, 4 to 5 hours. Now the results with the same sensitivity and specificity can be achieved within 4 to 5 hours. So I hope within 3 to 6 months, at least I can assure you, I will be in a stage to at least release this sandwich ELISA and followed by LFA. These are the all kits which have been developed abroad, and they have different sensitivity, specificity at different point of time. You see, again, variations are there, but they are definitely better than the progesterone methods. And I told you, they are marketing these kits in India also. Sensitivity again 86. So I tell you, we are very near to whatever the global standards are there. So, and these are the situation of the LFA. If you see their performance, so I'm happy to share you. Yes, our LFA also working very nicely, and definitely we will come in this to you only when we can say yes, it is LFA is working around more than 85 and ELISA is having a more than 90% sensitivity, and both must have at least 93 to 95% specificity. Accuracy should always be more than 90%, 95%. Altogether, because I have tried to, because I have been given only 50 minutes, I made some slides, fast, but because the audience is very diverse in nature, so that's why accordingly, definitely these are our lab finding. I have told you what is happening at global level. Yes, we are very near to develop a direct sandwich ELISA, an LFA which can diagnose pregnancy at around days 17 to 35 days. Even I can say it may be 11 to 42 days earlier than the clinical method. If you say whatever window I have told you for the clinical as well as. So even in that, if we could reach to that situation, it will be a very happy note, at least as on date situation. So this is my motto, this is my goal, and I need your wishes, blessings, everything. We should develop it. So these are some abroad how they are, like if you see type of the kits which are available. Thank you one and all for giving me an opportunity, especially Dr. Sudesh and Dr. Rinku Sharma, because she my junior and she was in here. She was putting the messages at play. So I accepted your invitation. I tried to justify the topic to the extent possible, the time available, and the expertise I do have. This is the area since long I am working. I am really thankful to one and all, and especially all those trainees, participants. For someone it may be not of that much important, but as you are present, at least that was my focus. At least you should get some awareness. You should what type of the state of art as far as early pregnancy diagnosis in India as well as a global scenario is there. Thank you very much, one and all.
Thank you so much, sir, for familiarizing us with recent advances in early pregnancy diagnosis in bovines, which is the need of the hour as far as every veterinarian is concerned. And now let us address few queries raised by our participants. Sir, first question is, recently Madras High Court has said artificial insemination is animal abuse. What is your thoughts on it, sir?
Oh, now again, Dr. Gopinath, you are putting another Pandora box, because as a biologist, I will go in the favor of EI, but as a sociologist, as a reformer, those who are thinking about the social system, it may be a tool which may interfere the natural process. But if as a scientist, I believe EI is as good as other techniques are there. Otherwise, if you think in this way, I think the whole of the globe, nobody will be able to use any technology. All technology are either invasive, semi-invasive, they affect your body parts. So then if no doctor will touch you, no doctor will give any advice to you, how you will get the advice? So I don't want to say more than that, because this is a very controversial area. Our Honorable High Court has told something, we should analyze all those things accordingly. Reply should be given. Sir, what is the state of art as far as EI is concerned? What is the advantage of the EI is in case of, you see, if you think in that way, what about IVF in humans? Everything will be flop. So I mean, we should think in a very positive way, seeing all pros and cons, our system. We should not challenge our natural system. I believe in that. That you should not cut the tree. Even trees are being cut, and but we should plant the tree. So we should cut the tree. Same way, techniques are meant for using for the betterment of the whole fraternity, whole human system. So as a biologist, reproductive biologist, I will go say yes, it should be utilized judiciously. It should not be, I mean, but I think it is another area. You should organize another lecture, a person who is expert in this particular area. So, and moreover, I will be happy to have a question from this particular pair. Any question is most welcome, but I think these questions are a little controversial because our Honorable High Court has so told. So I think Dr. Sudesh, I am right, I think, if you agree.
Sir, thank you, sir. Then next question, sir.
Yeah.
For LFA, are you generating your in-house antibodies?
Yeah. Everything in house, everything laboratory man. Nothing is from commercial.
Then please tell the cost of kit going to be developed at IVRI for checking a single cow.
What?
What is the cost of the kit going to be developed?
Cost we have not worked out till now, but you say because costing it depends on many other factors. Like because we are working since long type of the projects involved, but definitely when it will come in the market, it will be a cheaper issue that are say cost effective. Because in animal system, if we are going to sell anything very costly, no buyer will be there. And you see the fate of all idols, they are charging 30,000, 40,000 rupees for a 80 number of the assay. So there our farmer cannot pay. But if LFA comes, I think based on the performance of the assay, because major issue is that how effective our assay are. If they will be effective, cost will be definitely cost effective, and I think there won't be an issue. I after 3 to 6 months, I have told you now. After 3 to 6 months, I will be able to at least tell what will be the normal cost for the assay. And but it won't be because everything is in house. We are not using anything. So for us, it is not a big question. Thank you very much.
Okay, sir. Then few more questions, sir. What about hemodynamics of uterine artery? Is there any role in PD of bovine?
Uterine artery is a major artery which supply the uterus, and you know, whatever the blood circulation goes in these areas, it is basically. I am not ultrasonologist. We have done some work, but now people are using blood supply to the uterine artery as well as to ovarian system. They are using as a diagnostic tool. You have to correlate it. This is not because once you are taking the fetus, when you are seeing the fetus, why do you think others? In ultrasonography, if you are diagnosing the pregnancy, you see the conceptus, na? That is a major issue. If you are able to diagnose, why to see the other things? You can correlate all those parameters, no issue.
Then are you using monoclonal antibodies?
No, I am not using monoclonal.
Then why ELISA kits for P4 estimation give inconsistent result without any changes in cyclic, anestrus and pregnant animals?
Again, can you repeat
why ELISA kits for P4 estimation give inconsistent result without any changes in cyclic, anestrus and pregnant animals?
No. I mean, you see all those progesterone kits which are available. What they measure? It is a measuring of the progesterone. So if the progesterone is in optimum concentration, which is within the domain of that particular kit, and stress means there is no CL, so you won't get anything. It will be always less than 1 nanogram per ml. So there is no question. That's what I'm telling. For non-pregnancy, there is no question at all. Question comes when the pregnancy is there. Then another question is how early it is. Third question is that what is the values are there. If you are doing, if you do the same progesterone at day 45, 50, no issue. Problem is that how early you want. Everybody is worried about early, and all error comes when you go for early. If you think about dual 50, no issue. Animal is pregnant, and the progesterone will be higher. But what is the significance of such type of the assay? Doing 3 months, 4 months, 6 months, 7 months. Okay. By the time animal starts showing itself the symptoms. If an animal, if after 5-6 months, many persons they know they are animal going to be pregnant. So question comes how early, so that you can take all measures earliest. If it is non-pregnant, you can go for the management of the non-pregnant animal. If it is a pregnant, even pregnant animal require a lot of maintenance work. Okay, so accordingly, that's why kids meant for something else. We are trying to use for something else. Okay, because progesterone is not a specific substance which a conceptor secrete. Okay, so that's why that errors are there. For progesterone, there are many other issues. Things are there. Many conditions are there where progesterones are higher. That's why this type of results are coming.
Okay, sir. Thank you so much, sir. I can see many messages congratulating sir for your informative lecture.
Okay.
And once again, sir, thank you for enlightening us with a glimpse of researches going on around the world as well as the ongoing research in your laboratory as well. I hope our participants would be benefited from the data you shared with us for their future research as well as their ventures.
Thank you, Dr. David. I will definitely, I will have one word from Dr. Sudesh. After only. That's up, please. Are you mute? I think, Dr. Sudesh. I think, are you? Yeah.
Sir, please unmute, sir.
Dr. Sudesh, I think you are on. Please unmute yourself. Not audible. Dr. David Gopinath.
Yes, sir. Yes, sir. I will communicate him.
Communicate. I think I am not able
Yes,
to.
sir. Yes, yes.
Presentation which you gave to
Yes, sir.
participants, I think it's more informative. I do get some benefited on it also, which I was not doing earlier. Really, it's a very elaborative presentation. Thank you very much.
Thank you. That's how I couldn't hear much because you are mute. But I would like to hear something from you. I'm really happy to one and all, and Dr. Rinku, Dr. Nazir, and all those, Dr. Gopinath. You are trying at Palampur. It is a very happy note. At least our institute is taking a good lead. Thank you one and all. Thank you one. Thank you very much.
Over to Majid.
Yeah, should we leave now? Dr. Gopinath, should we leave now? Dr. Majid?